I'm an epidemiologist, pharmacoepidemiologist I've done quite a few studies on looking at drug treatment effects, and a number of those have involved trial emulation. So I'm just going to talk a bit about that space today. So yeah, looking at designing observational studies to emulate randomized controlled trials. So I'm going to cover some of the background and sort of concepts and theories, and then hand over to Paris midway through to provide a sort of more detailed example of doing this, and then Paris will hand back to me to finish off. So yeah, so this is the overview of the presentation. I'm going to talk about randomized controlled trials very briefly, then I'm going to talk about observational studies, and then emulation of hypothetical randomized controlled trials, and then emulation of actual randomized controlled trials. So first of all, just a bit about randomized controlled trials. So when you're studying drug treatment effects, evidence from well-performed randomized controlled trials is often considered to be the gold standard. So in a randomized controlled trial, you have people eligible to receive the new treatment, and they are randomized to either, in this example, treatment A or treatment B. And basically that randomization means that at the beginning of that trial, the groups are balanced in terms of their characteristics. So then if everything's been done properly, then any differences you see in the outcome you're studying, you can be confident that's to do with the treatment. So that's a big, obviously, advantage and big plus point of randomized controlled trials. But they do have some limitations. So a bit of a sad face here. So some key limitations is that they have very strict inclusion-exclusion criteria. And what this means is that people, for example, people with comorbidities, or for example, more severe disease, or actually milder disease, often can be excluded and won't be studied. They typically have a very short follow-up due to resource cost constraints. They can be underpowered to investigate subgroups, and it's not always feasible to do a randomized controlled trial for ethical or financial reasons. So then that's a very brief sort of overview of randomized controlled trials. Let's then just have a think about observational studies. So could observational analysis fill in the gaps from randomized controlled trials? So, for example, using very large databases of routinely collected electronic health records, such as you might have heard of the UK Clinical Practice Research Data Link. So the advantage of these types of studies is that you can have very large sample sizes, much larger than in a trial. You can include people with varied characteristics, so characteristics that wouldn't have been included in the trial. You can follow people up for a long time. It's basically as long as you've got data for, you can follow people up, and that can be years longer than in a clinical trial. You can include people taking treatments in real world settings. So that's in primary care, for example. You're actually looking at what people are being prescribed. And they are lower cost, quicker, and the ethics are often simpler as you're not intervening. So that's the smiley face for the observational analyses. But can they really be used to fill in the gaps in randomized controlled trials? Well, the key limitation is that there's obviously no randomization to the intervention. If it's routinely collected health data, say for primary care, the exposure is basically assigned to the participants in your study based upon decisions made by clinicians, and obviously the underlying condition that the patient has. So you have confounding that is difficult or challenging to overcome often. Okay, so there's no randomization, so you have confounding. But you also have other biases, and importantly, for stored electronic health record databases, these are time-related biases. And what I mean by that is that for a clinical trial where you're going out prospectively and recruiting people, you don't have information on what happens to them in the future, because that would be impossible. But in a stored database, you have, say, you can have, say, if you've got 30 years of records or whatever for the question you're looking at, you actually have information. If you design a study from a specific start point, which is quite early in that period, then you actually have all the information on follow-up already. And often there can be mistakes made where you're actually using future information in order to define or measure things, and that provides results that are very difficult to interpret and usually biased. So how about designing observational studies like randomized controlled trials? So the potential benefits of doing observational analysis like this have been recognized since the 1950s, and there's two articles there, one from 1953 and the other from 65, that actually are talking about that, that when doing observational work, it makes sense to look at how trials are being performed and trying to sort of design observational studies in the way that trials have been designed. But actually, obviously that's quite a long time ago, but I would say over the last decade, there's been a rapid increase in the number of observational studies that have been performed with key design aspects that have been based upon the design of randomized controlled trials. And that's what we're going to just look at today. So then let's go on to emulation of hypothetical randomized controlled trials. So you may have heard of something called the Target Trial Emulation Framework, or TTE framework. And what this involves is designing an observational study so that it explicitly emulates what's called a target trial. And what that target trial is basically, it's not an actual trial, it's a hypothetical randomized controlled trial that would have been conducted if you had no financial or ethical constraints. Okay. So this very well-known paper in the American Journal of Epidemiology in 2016 basically introduced the target trial emulation framework. So using big data to emulate a target trial when a randomized trial is not available. And in this paper, Ennan and Robbins talk about explicitly describing a hypothetical target trial. And as I just said, that's the trial you would like to conduct if it were feasible. You then emulate the target trial using the available observational data. And the target, so that paper outlines, and also you'll see it elsewhere in lots of other publications now and sort of commentaries around this, is the idea of a target trial protocol. So when you're doing your observational study design, before you do that, you design a hypothetical target trial using this protocol with these protocol elements. And so you think about eligibility, treatment strategies, assignment procedures, the follow-up period, the outcomes that you're looking at, what you're comparing, the causal contrast of interest, and an analysis plan. And I'm just going to go briefly through each of these, but as I say, there is a lot of literature on this out there already. So what you would think about is, before you start your observational analysis, you think about this hypothetical trial. So who would be eligible for that trial? What's their age? Are you going to restrict based on past health conditions, and restrictions based on prior use of the treatment of interest, for example? In your hypothetical trial, how would it work? Then in your hypothetical trial, you'd also think about the treatment strategies. So what treatment groups would individuals be randomized in that hypothetical trial? So what's the dose and the regimen of the active treatments? Is there a no treatment group? How is that defined? Then you'd think about, in your hypothetical trial, how are you assigning people to the treatments? How are the different treatments assigned? If you're emulating, it's worth thinking that there's no blinding possible. So even at that stage, you're starting to think, okay, well, this is my hypothetical target trial, but obviously when you're writing out the emulation, thinking about the emulation of that, then blinding wouldn't be possible, but it is in trial. Then for your hypothetical trial, you think about your follow-up period. So what is the start date that you would so what's the randomization time period? How long would you follow participants up in this trial? And what events would define end of follow-up? You think about the outcome then in your hypothetical trial, what outcomes and how are they measured? In terms of the contrast of interest, the causal contrast of interest, is it going to be intention to treat, so the effect of assigning somebody to the treatment, or is it per protocol, the effect of whatever they end up following? And then your analysis plan, actually in the target trial protocol, I'm getting ahead of myself here, but in the target trial protocol, you would, where you're outlining the trial, you would just be saying, okay, if it was a trial and it was randomized, then this is how I would do the analysis, which would be relatively simple if it is how you would do it for a trial. But actually, when you're thinking about the emulation in observational data, there then might be some more complicated analyses that you need to apply that in order to account for the fact that there hasn't been randomization. And that could be multivariable regression, propensity score, weighting or adjustment or matching. And then there are more advanced methods that you need to think about if you think that time-dependent confounding is a problem for your analysis. And the target trial framework doesn't mean that you can just follow it and you won't need to have a lot of careful thought about all the elements of your observation analysis, and that includes input from statisticians on your analysis plan. That all applies, but still applies. But the target trial protocol is trying to set out a framework to help you put your ideas together before you start implementing everything in your observational analysis. So this is actually the example protocol that Enan and Robbins had. I'm not going to go through this, but you'll be able to see that in their paper. They had their protocol with those elements, and it was related to the effect of postmenopausal hormone therapy on the 5-year risk of breast cancer. There's lots of examples out there. Here's another one, a more recent paper, which is a really nice paper that was in the BMJ earlier this year, looking at it was a target trial looking at second-line oral anti-diabetic treatments among people with type 2 diabetes. And I think what I'm just highlighting here is that they had a supplementary table that had their target trial protocol. So you can see on the left-hand side, it says target trial. I've just included 2 elements of the protocol, but if you look in this paper at the supplementary data, it has each element of the target trial protocol is outlined. Okay, so this is the ideal trial. And then what they've got is another column which has the emulation. So that is how it's going to be emulated within the observational data that they were using, which I think was CPRD. And that's really important, so that you outline your target trial and then you outline you think about how is it going to be emulated within the data that you've selected. So what's the point in doing this? So if you explicitly emulate a target trial, the key points are that this can really help clarify the research question. And I've thought, working a little bit in this area and talking to other people who've worked a little bit in this area and people who haven't yet done this type of study, some people say, well, isn't defining a target trial, isn't that a hypothetical target trial, isn't that just good epidemiology? And you could say, well, kind of like strobe or record PE are kind of all similar, like sort of guidances for performing studies. But I think the key point with the target trial emulation framework is that it really helps you think about the research question, because you're thinking about it as though it's a trial. That's the first thing. And I think the second thing is that it's very well aligned to using stored data, so big data, and it helps you avoid common design pitfalls when analyzing the data that you already have. And as I say, that's particularly related to defining time zero in your observational analysis and thinking about time-dependent covariates. And there's a very nice commentary piece here, which goes into some of these thinkings a bit more about why it's beneficial to specify a target trial if you're doing an observational analysis. So the target trial emulation framework has been applied very frequently and is increasing in observational analysis. So there's been very rapid uptake. This systematic review came out last year, And what it is, it says it's reporting of observational studies explicitly aiming to emulate randomized trials. Actually, it's a systematic review that specifically looks at observational studies that applied the target trial emulation framework. So that's quite a specific look at this. And it included 200 studies, and there were lots more that were excluded, which did similar things. So there's a lot of people now doing this type of work and applying the target trial emulation framework. And then there's another paper here that's quite a nice paper in the BMJ, and this is a sort of fast facts BMJ, which is about target trial emulation, about applying principles of randomized trials. And this is very easy to read, run through of what I'm sort of talking about now. But I was quite interested that in the conclusion to this, it said that observational studies are susceptible confounding regardless of the study design. So that's a really important point to remember. It doesn't matter if you're using a target trial emulation framework, there's still confounding to be considered. Then they go on to say, however, the emulation of a target trial using observational data will yield the same effect estimate as that of a randomized trial if the emulation is successful. And I thought about this comment at this sort of final sentence quite a lot, and it made me sort of think about, well, that sounds great, but it's still, how do you know that you've actually successfully emulated your hypothetical randomized controlled trial? And what I think there's still this enduring uncertainty. So if you're emulating a hypothetical target trial, there's a central challenge of all observational non-intervention analysis remains, which is, how do we know whether we've estimated results that are comparable to those for a randomized controlled trial of the same question? So basically, how does a research team know that they have performed a successful emulation, as was mentioned in that article? So now I'm just going to go on to the final bit of the presentation, which is around emulation of actual randomized controlled trials. So emulation of an actual randomized controlled trial. So to deal with that enduring uncertainty that I talked about when you're emulating a hypothetical target trial, so one approach is you still don't know if you've actually answered the question of the randomized controlled trial would have answered. So one approach is basically to take an actual existing randomized controlled trial as the target trial on which your observational study design is based, and then compare the results that you get with those of that same randomized controlled trial. And this step is known as benchmarking. And so there's an article there by De Harbour and colleagues, which is about the idea of benchmarking observational studies against randomized trials. But then it's like, so why would you bother doing this again? Why bother emulating an existing randomized controlled trial? If somebody has already performed a trial on the question you're interested in, why would you bother emulating that trial? So one key motivation is to increase the confidence in the methods and the data that you're using in your observation analysis when you subsequently extend that analysis to groups that are not included or underrepresented in the existing randomized controlled trial. So you remember at the beginning of the conversation, at the beginning of the presentation, I was talking about the fact that randomized controlled trials have very limited populations, and actually the people that use medicines in primary and secondary care are very different often to those in the trial. So what you can do is you can emulate an existing randomized controlled trial and observational data. You can benchmark to see if you get the same results, and that gives you an added level of confidence in your results. And what you can then do is extend your analysis to groups that wouldn't have been included in the randomized controlled trial. So the sort of procedure for doing that is then of emulating an actual RCT followed by extension. So typically what you might do is you would take the protocol of an actual existing randomized controlled trial, either published, usually the published version has the protocol attached, or you can contact the sponsor if you need further detail. You would then come up with a protocol for emulating that RCT and observational data. You would do the RCT emulation and in observational data and compare your results against the actual randomized controlled trial. And then you could then go on to extend your approach by including people who are excluded or underrepresented in that randomized controlled trial, or for whatever, for a longer follow-up, to answer these types of questions. So in the literature, there's more and more literature around this, and the actual existing randomized controlled trial has been referred to as various things when you're doing this type of approach. So you'll see index trial, reference trial, and origin trial have all been used. That's fairly typical for epidemiology. There'll be lots of, often there's multiple terms for the same thing, which can be confusing, but we're going to, just for the rest of this presentation, use the term reference trial. So I'm just going to give an example then of studies that have done this. So in this analysis by Matthews and colleagues, they benchmarked they were looking at coronary thrombus aspiration. What they did, they looked at a trial called the TASTE trial, which found no difference in the risk of cardiovascular outcomes by 1 year, with or without thrombus aspiration among people with ST elevation, a specific type of myocardial infarction. And what they did, they emulated, so they actually had they just emulated a target trial that was similar to the TASTE trial. So they defined a target, a hypothetical target trial that was similar to the TASTE trial. And what they did is they performed the analysis in observational data. They then benchmarked the observational results against the TASTE trial, and they found they got very similar results. What they did is they then said, okay, we're now going to look, the trial didn't, I think the trial was just for 1 year, but they extended the analysis to 3 years and looked at underrepresented subpopulations, which is, for example, those with diabetes. And what they found was similar effects to the 1 year analysis in the trial. So by doing it in that way, they're providing confidence in that these groups can be treated in a similar way as to those studied in the trial. So example 2, I'm going to hand over to Paris, who's going to go into some detail about a study she led that she's going to talk about.
Great, thanks, Kevin. Did you want to put yourself on mute just so I can hear myself?
Yeah.
So the reference trial here was on Target, which was a large global trial, which compared dual therapy versus ACE inhibitors alone. And it had 2 objectives, but the objective I'm going to focus on was assessing the superiority of dual treatment with telmisartan and ramipril, so an ARB and ACE inhibitor versus ramipril alone. And the trial was conducted in people aged over 55 with a cardiovascular diagnosis or high-risk diabetes, and it excluded people with heart failure. Next bit, please. Thank you. So the cohort included just over 17,000 participants, and 21% of those had chronic kidney disease. It had a primary composite cardiovascular outcome, which included cardiovascular death, MI, hospitalization for heart failure or stroke, and it also included a renal outcome. And for the main primary outcome, there was no evidence that dual therapy was superior to ACE inhibitors alone. And it actually showed that dual therapy was associated with an increased risk of adverse events. Next slide, please. So from the results of this trial, in conjunction with other trials, it led to an end of recommendations of dual therapy treatment. But there did remain some belief that dual therapy could still be effective in people with chronic kidney disease. But for some of the reasons that Kevin described previously, it's unlikely that a trial would be conducted in this population group. So this is where reference triangulation methods could be useful to study these questions. So the aims of this study were to emulate the design and benchmark the findings against the on-target trial before extending the analysis to explore the treatment effects and risk in patients with chronic kidney disease. Next slide, please. Thanks. So to do this, we used data from the Clinical Practice Research Data Link, and we selected all people prescribed an ACE inhibitor or an ARB between our study period, which was from 2001 to 2019. And then we went on to apply the on-target trial criteria. And to ensure that we ended up with a larger cohort of people with chronic kidney disease, we removed one of the exclusion criteria that was that previously excluded people with elevated creatinine. And then this resulted in an ACE inhibitor trial eligible cohort and a dual therapy trial eligible cohort. And we use propensity score methods to address confounding and achieve balance between the two exposure groups. So in routine care, typically people who start dual therapy don't receive these prescriptions at the same time. So to emulate this in our cohort, we created an operational definition of dual users. And this was that people had to have overlapping prescriptions for the two medications and receive a subsequent prescription for the first agent after the second prescription for the second agent. And then follow-up was then started from the date this definition was met. But we did recognise that this could introduce some survivor bias. So we looked at two alternate definitions in sensitivity analyses. Next slide, please. So just some baseline characteristics. So from our propensity score weighted analysis cohort, we had over 340,000 participants, so much larger than the on-target trial. And as you can see, the cohort is balanced between the two exposure groups in our analysis cohort, but the characteristics do differ to those in the trial. So our population was slightly older, we had a larger proportion of people with CKD and a more even distribution of females. And because of this, and the way this is designed, this then enabled us to have a more diverse cohort, more representative of the UK population receiving these medications, which allowed us to extend the analysis to those trial underrepresented groups. Next slide, please. So for the benchmarking step, so in our larger population, for the primary composite cardiovascular outcome that was studied in the trial, we found a hazard ratio of 0.98 and a confidence interval between 0.93 and 1.03. And this met our pre-specified validation criteria. So we were confident that our results were similar to the trial, and then we could then move on to extend the results. So to look at the trial underrepresented groups, we used the same analysis cohort, but we fitted an interaction term between chronic kidney disease and treatment. And in addition to the composite primary outcome studied in the trial, we also looked at a renal outcome, which was different to that was studied in the trial, but was of more clinical relevance. So that was a composite of a more than 50% reduction in GFR or development of end-stage kidney disease. And we found no evidence of treatment effect heterogeneity by CKD status at baseline. For the cardiovascular outcome, we found similar treatment effects between dual users and ACE inhibitor users, which was consistent with the trial findings. And then when we went on to look at the renal outcome, which was the main outcome of interest in relation to what prior belief was, we actually found that dual therapy was associated with an increased risk of renal outcomes, which was observed in those with and without CKD at baseline. So the conclusion that we found here was that this study provides evidence against that prior hypothesis that dual therapy could provide benefits among high-risk cardiovascular patients with CKD, which probably wouldn't have been conducted in a trial, but this method provided an opportunity to study this effect.
Great, thanks, Paris. So, yeah, after those two examples and that detailed example, where observational studies first of all emulated the trial, then benchmarked, then extended. I should also say that not all reference trial emulations actually that have been done so far actually then extend the analysis. So some of them that you'll see in the literature, they have omitted the extension step, and quite a well-known example of that is the RCT duplicate program. And this is an FDA-funded project to basically have a look to see what types of questions and therapeutic areas are able to be emulated in observational data. So they're basically trying to do as many as possible, but they're not extending currently to look in underrepresented or excluded groups. So there was a paper that came out last year with the results of 32 of these analyses, looking at 32 clinical trials. And so they emulated 32 RCTs, including 2 that were ongoing at the time. They actually had a common protocol template for each trial. And I think this is to do with the way that this group does their analyses, using a specific software approach. And it meant, obviously, the trials were across a wide range of therapeutic areas. So that may not be the optimal approach in terms of the trials can be very different across areas, and they use quite a similar protocol for each one. But they did deposit their protocols on clinicaltrials.gov after feasibility analysis, but before analysing any of the exposure outcome relationships, which obviously is commendable. So just some final thoughts on some of this. So I think one message from this presentation is that there's 2 types of sort of trial emulation that you'll see, and the target trial emulation framework, where you're emulating a hypothetical target trial, provides a sort of solid level of rigour to observation analysis of drug treatment effects. But I think it's important to be able to distinguish between observation analysis that's based upon a hypothetical target trial and observation analysis that's based upon and benchmarked against an actual randomized controlled trial, for example, often called a reference trial, and possibly extended to study trial excluded groups. And many of you may have seen this, so the nice real world evidence framework, which is really useful resource. If you look at the methods for real world studies and comparative effects, the target trial approach is sort of central to this. And the overall aim is to improve the quality of real world evidence informing our guidance. And I think they focus on the target trial approach as being important for this. But it's interesting that the examples that they give, so they give some examples of studies that have done this. And in fact, all 3 of them are studies that have emulated existing randomized controlled trials, rather than those that have emulated hypothetical target trial. But they don't, it's not actually, that distinction is not made in this guidance, and that was part of the reason for sort of starting to think a bit more about these differences in the 2 approaches. So obviously, though, it's not always possible to emulate an existing trial. For your question, so if you can't, if the trial doesn't exist already, and you think that trial would never be performed, and it is a trial, then you're going to have to, and you want to apply trial emulation approach, then emulation of a hypothetical trial is what you're going to have to go with. But if you are emulating an existing randomized controlled trial, then there are some additional considerations for reporting these types of emulations. For example, how do you describe the reference trial? What needs to go in there in your paper about that? How do you indicate that the benchmarking has been successful? Have you pre-specified your sensitivity analyses? I mean, obviously that's important for all observational studies, but I think if you're setting out to emulate an existing randomized controlled trial, perhaps that becomes more important to do. How to report the benchmarking results, exploration of any reasons for why benchmarking might have been unsuccessful, if that happened, and how to report extension of analyses to understudied or excluded groups. And then final thoughts. So the final thought is that observational studies based upon either a hypothetical target trial or an actual RCT are still susceptible to confounding. So, and you still need to carefully critique these types of studies. So it's not, and there's a paper there that's got some interesting points about that I would recommend having a look at. But it's like, even if obviously if you see that a study, if you're reviewing a study and it has, if it's emulated a hypothetical target trial or an actual target trial, it still needs critique, just as you would critique any other observational study. And yeah, that's it. Obviously, thanks to Paris and other people that have contributed to this presentation. So are listed here. And there is a preprint of a commentary that that myself and Anna Schultz worked on. There's actually a sort of more sophisticated version of that preprint is under review. We're responding to comments and should be hopefully published in the American Journal of FE fairly soon. Thank you very much.